Welcome to part 4 of our reproductive system lecture, and this is part 9 of our unit 9 lectures, our second to last lecture. We're going to talk about how fertilization occurs and how an embryo develops. And this is going to match, it's sort of going to jump around as far as which pages it matches, so I'll try to remember to mention each page as we go. Before we can talk about fertilization, we have to talk about how sperm are formed and how the eggs are formed. So we're going to start with how sperm are formed, and this starts on page 550 in your textbook. And the first stage is called spermatogenesis. So from birth until males reach puberty, they're going to be undergoing mitosis. And notice I put that in bold. Mitosis is the one where you're just making copies of cells just for growth or for healing. So this is going to grow the number of stem cells they have that are potentially going to make sperm. Those stem cells are called spermatogonia. And the reason I put 2n equals 46 here in the parentheses is because, remember, n is the number of chromosomes we have, but we have two sets. We got one from our mom and one from our dad. So these stem cells still have that complete set from mom and dad, or 2n, which is 46 chromosomes. So we have not chopped them down into half to pass them on yet, like we're going to need to do ultimately. And that's also why mitosis is bold here. So from birth to puberty, basically preparing these stem cells that are going to go on and make the sperm. Then at puberty, a hormone, we're going to refer to it as FSH, which stands for follicle stimulating hormone, is released, and that's released by the anterior portion of the pituitary gland, which we've heard from a lot. And when this hormone is released, it's going to trigger meiosis to start. Now, meiosis is the one where we're not just making an exact copy of the cell. Now, in the process of making more cells, we are cutting down the number of DNA into half. So you have this double setup here. Only one from each of those double pairs can get passed on. So we're only going to pass on 23 chromosomes from dad when we make the sperm. Okay, these are called spermatids. And they're almost there, but they're not functional as sperm yet. So they've got the genetic information part, they've got the right number of chromosomes, but they're not ready to go as far as sperm. So before that can happen, spermiogenesis needs to happen. So let's go back for a second. Spermatogenesis makes a spermatid. So this is getting the DNA information down to the right number. Then spermogenesis is going to make a sperm to make it functional as a sperm, and there's a picture of a functional sperm cell here. Okay, so the spermatids have a couple of problems. They're not motile, they're not ready to go. As your book says, they have too much excess baggage. Remember, their job is going to be to swim and to swim fast and to get to where they need to go before they die. And with all that extra stuff, they're not going to be able to. So basically, all that extra stuff, all that extra weight is stripped away. They grow a tail, and they're going to become very streamlined and have a very high metabolism to support all this fast swimming. They're going to need to make a lot of ATP. So they have three sections. Here's the head section, and then that's got two things going on. It contains the DNA, so all the genetic information is going to be stored right here. And then it's got this covering called an acrosome. That acrosome has enzymes. So when this sperm hopefully reaches an egg, it's going to need enzymes to help eat through the outer layer so that it can actually penetrate the egg and fertilization can occur. That's what those enzymes are going to help with. Then you have the mid piece you can see here. The mid piece is home to lots and lots of mitochondria. If you remember when we reviewed cells at the very beginning of the year, and hopefully you remember from biology, mitochondria are the organelle that make energy. So this is going to make all the ATP needed to support the swimming. And then there's the tail, and the tail contains lots of filaments that are going to actually do the swimming. Okay, now the sperm is ready to make the trip to go meet the egg before the egg is no longer viable and before the sperm dies. Okay, so we had spermatogenesis that's going to cut down the DNA information to half, and then we have spermiogenesis, which is going to make that sperm streamlined and ready to go. This is all occurring while that immature sperm, that spermatid, is traveling through that long length of the epididymis. Remember, we said that takes a really long time, and that's being moved along by peristalsis. It has time then to mature so that once it's actually ejaculated, it can swim on its own. Okay, so now we know how the sperm are made. Let's talk about how the eggs are made, and this is called oogenesis. Remember, that double O prefix is referring to the egg. Genesis refers to making something or creation. Okay, so this actually starts before birth. So when females are in the womb developing, they're actually starting this process. Okay, so the stem cells, in this case they're called oogonia, are undergoing, again, mitosis. They're just making lots and lots and lots of extras to prepare. And we call those primary oocytes. So they're still, with all 46, they've got their double set. Okay, and these are going to push their way into the ovarian tissue. So now they're going to be in the ovaries, ready to go. This is all, again, before this girl is even born. Okay, this is happening in the womb. About 2 million of these primary oocytes are made before birth. So that's all that girl, 2 million sounds like a lot, but that's all she's going to have. After birth, no more are made. Okay. Then between the time of puberty and menopause, that same hormone, the follicle stimulating hormone, is going to trigger a few follicles each month to sort of mature and go on. So now here's where meiosis comes in. As these follicles are going through the process, they're going to undergo meiosis, and they're going to get down to this one set number, this 23 number. Okay? So by the time the mature egg is released, by the time ovulation occurs, you have the right number of chromosomes going on. Then another hormone called luteinizing hormone is going to trigger ovulation. And notice here it said a few follicles. So a few are going to get ready, and whichever one is sort of most mature when this hormone hits is the one that ovulates. So in very few cases do 2 eggs get released at the same time. Usually it's one and the others just degrade. I don't really expect you to know the specific names here of the hormones, but just recognize that all of this is under the control of hormones. Okay. Unlike the sperm, ovum are really well stocked with nutrients. Their job is not really to swim anywhere. They're going to get moved through those fallopian tubes with the cilia and with the peristalsis, and then they're going to implant in the uterus. The sperm have to come and find them. So they're going to be stocked with nutrients to supply that ovum with what it needs. Okay, so now let's get to fertilization. When do these guys actually meet? First, let's talk about viability or lifetime. How long can these gametes survive? The ovum can only survive somewhere between 12 and 24 hours from the time of ovulation. So once they're released from the ovaries, they've got a day at best. And the sperm can only usually live about 24 to 48 hours from the time they're ejaculated. So they have to swim and find that viable ovum within a 1 to 2 day space. And that ovum has to have been ovulated within that time of a 1 day space. So there's about a 3-day window for fertilization to occur. If sex occurs within a 3-day window of the time that a woman is ovulating, there is a very good likelihood that she will end up pregnant, because both the egg and the sperm will still be viable when they meet. Even though there's millions of sperm ejaculated at one time, only a few hundred of them actually complete the full journey up through the duct system of the female, through the fallopian tubes, to meet this viable ovum. And I have a link later in this presentation where you can watch a YouTube video that talks about a lot of the reasons that not most of the sperm make it. So if you're wondering why there needs to be millions in just a couple milliliters of semen, this is why. Most of them don't make it, so it gives them a better chance to have so many. Okay, once they do reach the ovum, there's going to be lots and lots of sperm all coming toward this ovum, and the enzymes from those acrosomes on the head of the sperm are going to start weakening that follicle that's around the ovum, the cells around it. And once this happens, one sperm will make it through first and make contact with the cell membrane receptors, the things surrounding that ovum cell. And they're going to recognize that a sperm has gotten through, and they're going to cause some changes so that no other sperm can enter. Okay, so once fertilization occurs, no more sperm can enter. And usually, your book talks a little bit about this, that one sperm that makes it through is not the sperms that did all the work of weakening the outside. I know some of you are probably saying, well, what about twins? Okay, there's 2 types of twins, and we're not really going to go into it in the lecture, but just briefly, there's identical twins. That occurs after fertilization. So one egg is met by one sperm, and then that zygote, that new cell, splits into 2. And that's why they have exactly the same genetic information and look so much alike. There can also be fraternal twins, where 2 separate eggs get fertilized by 2 separate sperm. It just so happens at the same time. So when that ovulation occurs, 2 eggs get released, which is rare. and then 2 sperm, they happen to both get fertilized, and they'll both implant in the uterus. In that case, they're not really any more genetically similar than siblings might be. So they don't typically look alike like identical twins do. Okay, and like I mentioned, I think a few minutes ago there, once the sperm does enter the egg, now that half set of DNA from the sperm has met with that half set of DNA from the egg. Now we're back to a full set, and that first new cell of that human is called a zygote. Okay, on this slide, I'm briefly going to talk about some of the key points along the stages of that embryo that's developing. So now we've got the genetic information from mom and dad together into the zygote. The zygote's going to carry on, and it's going to start multiplying through mitosis and eventually build into the infant that will be born. I'm not going to go into a lot of detail because we're so pressed on time, so I don't expect you even to know kind of all of the vocabulary I talk about here, but I'm going to include some information in case you're interested, and there's a lot more detail in your textbook. So first of all, that zygote, remember, fertilization happens in the fallopian tube, so we're not to the uterus yet, so it needs to continue its trip on and end up in the uterus. Okay, on the way, it's going to multiply a little bit, so some mitosis is going to occur, and we're going to make copies of that first cell. Okay, now it's called a morula, and it's about 16 cells big. It reaches the uterus at this point, and it's actually going to float for a little while in some secretions that have some nutrients in it. So that endometrium isn't quite ready to be implanted, and it's going to kind of take its time. When it reaches about 100 cells, it's called a blastocyte, and at that point, it can implant into the endometrium. And remember, there's lots of blood flow there and lots of nutrients, and so that's going to be a good place for it to get supplied with what it needs. A hormone called HCG is going to be released at this point. That's sort of triggered when it's implanted, and this is particularly important because remember, up until then, this woman has been going through a cycle of 2 different hormones, the estrogen and the progesterone, going back and forth depending where she is in her cycle. We want to stop that. Okay, now we want to sort of reset, not to be cycling through, but to be focused on letting this embryo develop. Then the placenta, which is going to be what delivers nutrients to the baby and what filters out waste, is going to start to develop. The umbilical cord, which connects the placenta and the baby, and the amniotic sac, which surrounds the baby, are all going to begin to develop. At 8 weeks, we call it a fetus. And this is the point at which the groundwork has been laid for all the major organ systems.
If it has a 0 here, they didn't give a cell phone number.
Okay.
This is the front of all the